Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the course Experimental Biotechnology, we are discussing about the different types of techniques. So in this context, so far what we have discussed until the live session 1, we have discussed about the standard operating procedures, and we have discussed about the precautions and other things what you have to do. And in the subsequent modules, I think we have also discussed about the different types of techniques, right? So what we have discussed so far in this course, so live session we are doing, live session 1 we are doing after I think the module 3 or 4. So in the module 1, mostly we have discussed about the operation of the instruments, and we have also discussed about the precautions and the standard operating procedures, like what will happen if you get the if you are working with the radioactivity and all that. And then we have also discussed about the basic principle and as well as the operation of some of the instruments. So in this we have discussed about the pipette man, we have discussed about the pH meter, and we have discussed about the water distillation unit. And I have tried that wherever it is possible, we have given you a demo also, right? How you can be able to do the pipetting. So I have given you a demo also. What are the precautions you should take? What is the correct way of doing the pipetting and all that? And so we have also discussed about the laminar flow or laminar bench. Then we have also discussed about the other minor instruments like the centrifuge and all that. Then module 2, we discuss about the electrophoresis, and within the electrophoresis we discuss about the basic principle of the electrophoresis. We discuss about the how to running of the gel, so running of the different types of gels, different types of phase gels, right? So we discuss about running the native gel and the denatured gel like the SDS page. Then we also discuss about the we have also discussed about the staining procedures, and at the end, in this particular course, we have also discussed about the troubleshooting. So there are specific aspects what we have discussed. And then in module 3, we discussed about the application of the page, and we have discussed some of the experiments related to the electrophoresis, where we have taken some of the classical questions, how you can be able to use and exploit the electrophoresis. So we have discussed about the DNA protein interactions and so on. So if this is all a broad summary what we have discussed. In detail anyway, we have discussed the content, and wherever it is possible, I have tried that I should show you a demo so that you will get a virtual experience. And if the time will permits, some of you probably can visit to my lab for summer internship, and that actually is, I think there is a complete program by the NPTEL office that you can actually be able to choose my laboratory for the summer internship, where they are actually going to give you the fellowship and all other kinds of things. So all these details are anyway available on the NPTEL website, and you can actually be able to consult the NPTEL office for more detail. So with this brief discussion or the summary of whatever we have discussed, I will suggest that we should start asking the questions, and we should further discuss about the different types of you know, different aspects, and whatever you have the doubts, we can also keep that. So if you have any questions, you can ask me. So do you have any question? Lavanya, we'll start with you. Do you have any question or anything you could not be able to understand? We can discuss.
Yes, sir. Hello?
Yes, yes. Can you introduce first yourself? So
Yes,
that,
sir.
yeah.
Yes, sir. In assignment 4, can you just explain the entropy, enthalpy, and the free energy of the system, sir? Like, what is the difference?
I think entropy, enthalpy, and free energy of the system I can explain you. Okay, but it is better if you could actually be able to see some specific course on that, because it is actually, these are very big topics to actually elaborate and tell you in 5 minutes.
Okay, I understand.
So better you see some chemical engineering courses where they probably will actually explain in detail about these terminologies. So these are thermodynamics parameters which are very difficult to explain in 1 or 2 minutes.
Okay, sir, sure.
And I think you have so many doubts, or I think some questions related with the assignments also, right?
Yes, sir.
So I could not understand what you are talking about. But like you said, assignment 2, question number 7, explained in assignment 3. What does that mean?
Yes, sir. Actually, these set of questions in assignment 2 have been explained in the lecture series of the assignment 3 or module 3. So, we are not able to get the answers and submit it for that assignment, sir. So, it is answered in the preceding modules. That's what I meant, sir.
Okay. But I think that is question number 7, right?
Yes, sir. If you want exactly, I can also tell you in which part of the next module it is explained. But since when we are submitting, for example, when we are submitting assignment 4, we normally listen to the module assignment 4.
Actually, that happens sometime because some of the topics are not very confined to particular pinpoint. Okay, so because number 3 and 4 both are related to electrophoresis, there could be this kind of thing could happen actually. But so can you specifically say question number 7? What is question number 7?
Right, 1 minute. Question number 7 is that after running STS page for protein sample, protein band appears as double band. What is this problem can be solved by? That is the question, sir. And the smiley band. This is also explained in the next module.
Okay, got it. Yeah, this happens because sometime because the topics are pretty overlapping. So that's sometime it could happen. We'll try to keep it that will confine it to only to that module only. But
Sure, sir.
yeah, I understand that now. Okay. So same is true for this, right? Molecular weight of
Yes,
a protein
sir.
determined from SDS page will be inaccurate if all these are from module 4, module 3.
Yes, sir.
So that is acha. Okay, fine. I'll cross verify. Okay.
Sure,
Anyway.
sir. Sure, sir.
Any other query you have?
Sir,
Apart from that, what is the charge of the methylene blue, sir? Like when it's
Charge
in
on the methylene blue is positive.
positive.
Charge
So
on
in gel
the
yes, sir.
charge on the bromophenols blue is negative.
Okay, so
That's
so
why
in gel electrophoresis,
that's why you can use both of these dyes combination in the horizontal page, and they will go to the respective electrodes. So you can be able to monitor, or you can use them as a tracking dye.
okay, sir. In gel electrophoresis, it will move
Is it clear
towards.
now?
Yes, sir.
Yeah. So methylene blue is positively charged, so it will go towards the negative electrodes, and the other one is will go towards the positive electrode. So in a standard setting, you have negatively charged protein, and that's why we are using the bromophenols blue
Okay, sir.
as a tracking dye. But when you are working with the horizontal page, you need to track the other side also, right? So
Yes.
that's why we use the methylene blue.
Okay, sir. And in assignment 4, we should get some notes on thin layer chromatography, sir. So, like, where can I seek the
In assignment 4, what is that?
thin layer chromatography is like where can I refer for that, sir? Module
We have
4.
not discussed about the thin layer chromatography.
No, sir. That is for ion exchange, column chromatography, and other things have been discussed. But thin layer chromatography questions are there, 10 and 11. So I just need some reference points for that so that I can answer those questions.
Okay. So assignment 4, I think still we have time, right?
Yes, sir. This Wednesday is the last submission dates. We have time.
Okay. So what I'll do is in the discussion forum, probably I will post the content. I will post
Sure,
the
sir.
material related to thin layer chromatography.
Sure, sir. Thank you so much, sir.
Yeah. Anybody else?
hello, sir. My name. Hello, sir. My name is Mayank, and I'm a MSc Biotechnology student. So I just conducted an experiment, and in which I treat plasmid with topoisomerase, and I run
Your
the.
voice is not clear. Your voice is not clear. Can you repeat now?
Sir, is it clear now?
Yes.
Hello. Sir, my name is Mayank, and I am a MSc Biotechnological student. And I just conduct experiment in which I treat plasmid with topoisomerase, and then I run with in the gel electrophoresis. So one of them is control, and one of them is after the treatment of topoisomerase. But there is no difference between those two bands. I mean, topoisomerase activity is not shown in the gel. What could be the reason, sir?
See, until I can't see your gel and until I can't know the 100% everything, right? How I can comment on that? See, you're not getting a topoisomerase activity. Could be because of topoisomerase itself is not active. So how we can actually be able to cross verify that your protein is functionally active? Number one, if I want to sort out this issue, right, that topoisomerase is not doing the, it's not causing the supercoiling, right? That is what your activity is, right?
Yes, sir.
What I'm saying is, if I could understand correctly, that if I run the topoisomerase with plasmid, I should see a shift in the plasmid bank.
Yes, sir. I should see a shift in
You
the
should
plasmid
see a
band.
shift in the plasmid band because plasmid is going to be supercoiled, and the supercoiled DNA will migrate differentially compared to the plasmid DNA, right?
Yes, sir.
So, number one question is: did you purify the plasmid with kit?
Sir, we didn't use the kit, but we used different buffers and we use
So,
different
did you use the
enzymes.
alkaline lysis method? Did you use the alkaline lysis method, or did you use the
Alkaline
standard
lysis,
column-based kit?
alkaline lysis method.
Okay, so then it is fine, because if you use the kit actually, then the topoisomerase activity will not be there, because the plasmid is going to be supercoiled plasmid. Mostly these kit-based purification gives supercoiled DNA. They don't give the other form of the DNA. Whereas in the alkaline lysis method, you will get the plasmid, okay, the uncoiled plasmid also. The second thing, what you should actually verify, that the topoisomerase what you are using is actually binding the DNA or not, because it has to bind the DNA,
Yes, sir.
right? So if it binds to DNA, then only it can actually going to generate the supercoiling. So these are the some of the steps probably you have to cross verify before you can actually conclude that the topoisomerase is activity. You are not getting the topoisomerase activity. So topoisomerase, first thing I will do is I will run it on a gel filtration column. You have overexpressed this topoisomerase, right?
No, sir. There's no shift like the
No.
control band.
See, listen. You have to, from what is the source of topoisomerase? You have purified or you have purchased topoisomerase?
Purchased topoisomerase.
Purchased topoisomerase. So, did the company from where you have purchased the topoisomerase gave you any quality data? Quality data in terms of, like for example, if I have sufficient topoisomerase, I should do a small gel filtration column, so that I will know whether my protein is native or not. Number two, I should do a small CD spectra to see that whether I have the secondary structures or not. If any of these things are actually not matching with the standard CD spectra or gel filtration column, gel filtration parameters of the the reported structure, right? Then you, whatever the protein you have is not functionally active. So if it is not active, you will not get the plasmid supercoiling activity. The third, anyway, you can
Okay.
easily do a binding study. See, until
Yes,
the
sir.
topoisomerase is not showing you the binding study, it will not show you the other thing. And the fourth is, I think I remember topoisomerase has a ATPase activity also, right? Because it requires the ATP for this activity also.
Yes, sir.
So it may have a ATPase activity. So that ATPase activity goes up when you add the DNA. So ATPase activity, measuring the ATPase activity is very easy. Until you don't do this troubleshooting, it is very difficult to figure out where your things are going wrong. You got it?
Yes, sir. I got it.
Yeah. The other way of how you can be able to even cross verify this is you just prepare a nuclear extract. Okay. Right.
Yes, sir. Okay.
You take some eukaryotic cell, right? Isolate the nucleus, prepare a nuclear extract, okay, and use that as a source of topoisomerase?
Yes, sir. I will do that
So
also.
it will not give you the clear-cut answer, but it will be going to work as a positive control. So if you add the plasmid to this, the topoisomerase, what is present in the nuclear extract, is going to give you the supercoiling.
Okay, sir.
So that will sort out whether you are making any mistake in terms of preparing the buffers. The pH could be one of the critical parameter. So if all these things are if your assay is working with the nuclear extract, then you can then you can work on the topoisomerase, the what you have purchased from the company.
Okay.
Got it? So you're done, right?
Yes, sir. Clear.
Yeah. So anybody else want to ask anything?
Hello, sir. Hello.
Okay.
You are able to hear me, sir?
Yeah. You can ask question.
Yeah. Sir, suppose I want to know about this purification of. Suppose now I have an extract, sir, containing many different proteins. Example from beans or some plant material. Now I want to isolate one particular protein. For example, lectin. So you were telling about salting in and salting out. And will you be telling about this dialysis bag? Because I want to know. I read one article where they say molecular weight cutoff, you have to use a dialysis bag to purify if you want to isolate lectin from that. So in your subsequent lectures, will you be telling about that, sir? Or do you have to?
There's a lot of noise coming from your side. I think yeah. So you want to isolate a protein from a abstract.
Yes, the particular protein. Hello.
Okay. Yeah. Then
I want to isolate a particular protein. I want to isolate lectin. So when I read the paper about how to isolate the lectin, they were telling about some dialysis bag. You have to use dialysis bag, and then you're going to make use of salting in, salting out procedure. So with some particular molecular weight cutoff. So will you be explaining to us in your subsequent lectures?
dialysis I have not discussed, but I think salting in, salting out we are going to discuss. I think probably in module 4. I think.
Okay. Then I will wait for that, sir. All your lectures are very nice and very easy to understand, and you start with the basic and you take up to the higher level. Really very nice lectures. I want to tell at this point.
Okay. Okay. Fine. Anybody else want
Thank
to
you.
ask? Yeah. Okay. Thank you. Anybody else? This session is only for 1 hour. If you have any question, you have any doubts, queries, you can ask me.
Hello. Sir, is it audible?
Yes, Veena, you can ask.
Yes. Yes. Sir, I have a doubt regarding the addition of TAMED in SDS preparation.
So what is your background?
So we are plant physiologists.
What is?
Plant physiologist. Botany.
You're a MSc student.
Yes. No, sir. PhD.
Oh, you're a PhD student. Okay. Plant physiology is your topic.
Yes, sir. Yes.
Okay. Now ask me.
Okay. So, sir, actually, we were just doing a rough work of this SDS, initial work. And while I had added the TEMED around 0.3 microliter, just doing a trial, and I had added 0.3 microliters of TEMED, but
but it takes around 30 to 40 minutes to solidify, and sometimes it more. But then I increase the concentration, so I found that it solidified within a minutes. That even I didn't get a time to just cast the gel.
Okay, fine.
Okay, so my question is, what is the actual error? Is it the
See,
camera? Is it the,
okay. So your problem is that when you are adding the timid,
yes,
small amount, your polymerization is very slow.
yes,
Right?
yes.
Your problem is when you are adding more, the polymerization is instant. So you are actually not
It becomes yes.
getting enough time for.
Yes,
Right?
sir. Yes.
So tell me how much APS you are adding.
It is just 50 microliter.
50 microliter in 5 ml.
Yes, sir.
Okay, fine. So that is perfect. Okay. So what I will suggest, and that you can try out. Okay.
Yes.
Prepare all these recipe, right? Whatever we have said, right? You add the buffer, you add water, you add acrylamide, you add everything, and then what you do is you take how much, 2 microliter or 3 microliter timid, not more than that.
Okay, sir. Okay.
Add the timid first.
Add the timid first and then APS.
Yes. Add the timid first and then you add the APS. Mix it. For it, it will be done. The second point is your lab is air conditioned lab or the room temperature lab? It's like,
So
is it
it's
without
room
AC?
temperature. It's room temperature.
Yeah, room temperature lab. So probably even if now also you are getting a very high polymerization rate, what you can do is just keep your these buffers in 4 degree.
4 degree.
So that you will reduce the polymerization rate. Okay. Half an hour is a very late. Normally
Yes.
it should take 5 to 7 minutes or 10 minutes max.
Yes. Yes.
So the third thing what you can also try, which I won't suggest for SDS page, because otherwise sometimes people, I think we have discussed that in our course also, that you can use the riboflavin, but that is not which is going to sort out your problems. What you can do is you can work with the colder buffers, right? And that will take care of this accidents, whatever is happening, that it is getting polymerized very fast. And don't mix it very vigorously.
Okay.
Okay, because if you mix it very vigorously and your outside temperature is 40 or even more than that, then the polymerization will actually going to be very fast.
Okay.
Just mix it, pour it. That's it.
Okay.
And I think for pouring also you might be using the pipette, right?
Yes, sir. Yes, sir.
No need to use the pipette. Just directly tilt your apparatus, right?
Yes.
And pour it with the beaker itself.
Okay.
Because if it is more, see, by doing practice, I think you will do it very fast. Okay. So it will not be polymerized before it get into the plates.
Okay, sir.
Yeah.
Okay. Thank you, sir.
Yeah. Anybody else?
Hello, good afternoon, sir.
Yes.
Yeah, sir. Yes, sir, I'm a PhD student in biotechnology only. Sir, I wanted to ask you that according to assignment 2, protein kinase assay can be performed using the horizontal native gel electrophoresis. So, what is the possible what is the concept and what is the reason behind that? Can you please explain, sir?
Okay, let me share my screen and I think I'll explain it.
Okay.
And I will open my assignment 2 also. So,
yes.
Okay. Okay. Do you see this screen now?
Yes, sir.
Okay. So the question is very simple. You have a substrate A. You are going to add the ATP in the presence of a kinase, and that is going to generate this, right? And
Yes, sir.
if you are going to talk about the reversible, then it is going to be phosphatic. Okay. Along with this, you are also going to generate the ADP. Okay. So you are actually having the two species. One, A. If I want to monitor this, okay, I have to measure the molecules, number of molecules of this A phosphate species, right? So A phosphate is not going to be very, very different in terms of molecular weight, right? So the only thing what we can do is, because we have to separate the A from the A phosphate. And this is, I assume that this is actually a neutral molecule, okay? Or I can actually work in a pH where the A is going to be neutral. Okay, A could be a protein, A could be carbohydrate, A could be lipids, A could be anything. This one is going to be negatively charged. Okay. So if I run this mixture, okay, into a vertical native page, so that is the first option. Okay. If I run this on a vertical native page, that vertical native page will not be able to separate the A from the A phosphate, because the differences are going to be very little. Okay. In terms of M by Z ratio.
Okay.
Because A versus A phosphate, phosphate is not going to create very high amount of, in terms of charge differences. Okay. So that will not be very practically, it is not possible. SDS page, anyway, it is not possible under this condition. As long as you do the SDS page followed by the Western blotting, then it is possible, because then you are using additional probe which is actually going to detect. So you are going to use the antibody which is going to detect the A phosphate, right? So then it is possible. Otherwise, SDS page is actually going to give you the two bands which are going to be same, because SDS is going to neutralize whatever the negative charge you have on the A. So A will actually not going to have any difference in terms of migration if you're talking about the SDS page. Same is true for the native page also. Now 2D gel electrophoresis. 2D gel electrophoresis is can be used, okay, and but in terms of the 2D gel electrophoresis, what you're going to do, right? You are going to first run at the level of PI, right? So PI is going to be different for both of them. PI is going to be different.
Yes, sir.
PI is going to be different only if you assume that A is a protein actually. If it is a non-proteinaceous substances, forget about this, okay. So PI is how the PI is going to be different.
Different charge. Oh, achha, no.
Yeah, but these contributions are very little actually. You cannot do simply the 2D gel electrophoresis. The same is this is actually the same logic for SDS page also. If you do a 2D gel electrophoresis, A and A phosphate, they will actually going to be very close. They will be like this.
Okay.
Okay, so people actually cannot do simple a 2D gel electrophoresis and say, oh, this is the phosphate, this is the phosphorylated band, this is the unphosphorylated band, until they will actually going to follow with the antibody, or they can use some probe to say that this is phosphorylated, this is unphosphorylated. So further staining is required even if you do the 2D gel electrophoresis. Now let's talk about the horizontal gauge, which is actually the correct answer. Now horizontal page, what we said, okay, this. You are actually going to load the sample in the middle, right? This is going to be positively charged, this is going to be negatively charged, and I assume that A is neutral. You can assume A negatively charged or positively charged, but if I consider the A as neutral, so in this gel I have loaded the control reaction. In this gel, I have loaded the reaction where you want to measure. Okay. Now this is you're going to have A actually, A alone, right? Because you have removed the ATP from the reactions. So you're going to have two reactions. One is control reaction where you are going to have A alone. Then you are going to have the experimental reaction where you are going to have A plus ATP plus enzyme. So here you are going to have the enzyme alone. So there will be no phosphorylation. This is actually going to give you A phosphate. Okay. So in this one, you're going to have A phosphate. Now, I assume that A is neutral, right? So what will happen? If you develop this, right, what will happen is that you will have a band here only, and you are actually going to have the band which is somewhere here. So you're going to have this band which is corresponding to A, and you are going to have this band which is corresponding to A phosphate. Although even the differences between the A phosphate and A would be less in compared to the charge, you might have to run this for 1 round or 2 round or 3 rounds. So even if you run it for 3x time, okay, 3x means you are going to run tracking die from 1 to this side, that is called as 1x. So if you run the tracking die 3 times, which means you run the gel for 3 times, then that is called as 3x. So even if you run 3x or 4x, there will be a subtle difference between the A phosphate from the A. And that's how you can be able to measure the intensity of A phosphate from the A. And that's how this ratio is going to tell you A phosphate concentration versus A is going to tell you the enzyme activity. That how much percentage of the A is getting phosphorylated in a given time frame. Is it clear now?
Yes, understood. Understood, sir. Thank you so much for your explanation.
Yep.
Thank you, sir.
Anybody else want to ask? So we only have 10 minutes left to wind up the session. In case you have any query or you want to have any doubts, you can ask me. I think there are questions in the chat box. So I think there is a question in the chat box. In the chat box, the Selvam is asking, sir, why not we use directly the DNA to PCR? Another experiment, why we use directly DNA to PCR, and for other experiments, the DNA convert to cDNA, convert to messenger RNA. No, it's not correct. We normally use the directly DNA into the PCR reactions, but in the other reactions, we convert the messenger RNA into the cDNA, and then we use them in the other kinds of reactions, whether it's the PCR reactions or the other kinds of molecular biology reactions. So So we don't convert the DNA to cDNA actually. We convert the messenger RNA into cDNA, and then we use that as a source of template for other kinds of applications, whether the PCR or even for the cloning also. If you have any more question, you can drop that into the chat box also. So since you are not asking any question, and I assume that the topics, whatever we are covering in this particular course is it's clear to you and you don't have any doubts. Even if you have a doubt in the in future also, you can use the discussion forum, and it would be very nice for others also to follow the discussion forum, so that many times you could not we get that idea, but students are actually asking it. So it's a very good idea to discuss your concerns in the discussion forum. And yes, we are not having any more doubts or any more questions from the student side. I would like to conclude these sessions. And so what we have done, we have discussed about the some of the aspects, what are questions from your side. And so with this, I would like to conclude our live session one, and I'll be happy to answer your questions in the discussion forum. So please use the discussion forum for the fruitful discussion. And every day morning, I actually open the discussion forum. So you will get the instant answers, and hopefully that will help you to understand the topics nicely. So with this, let's conclude this session. Thank you for your attention. Bye-bye. Good evening.